Let us continue lecture 2 of session 2. What are the role of ion transporters in cellular pH regulation? Of course, cellular pH is an important one and it should be maintained within normal range for the cellular process to occur normally. So kada pH بقوله, when pH change, there will be change in the most cellular processes, even the enzyme. The enzyme can act in a specific pH. So pH regulation is very important. How pH regulated inside the cell? Regulation is mostly through cells buffering mechanisms. لأن cell can بفرق معناها كملا buffering mechanism معناها like phosphate buffer, protein buffer, and so on. Can buffering mechanism معناها, but when these buffering mechanism exceeded, how the cell regulate or control its pH كاتك pH أونا قران كهربا الصراحة وبفر كانش نانتواني شاكي كان شيبه. The transporter will work in this case. So how the transporter work? If there is intracellular acidity كاتك pH in our cell can be during low cellular pH, the cell should increase its pH toward normal. How it can do that? pH can be يعني H zora, hydrogen ion zora. So it should expel hydrogen ion out, or there will be inward movement of bicarbonate. So the cell can do one of these or both of these mechanisms in order to overcome the cellular acidity. What about the cellular basicity? When there is high cellular pH, it is opposed by expelling bicarbonate. كات bicarbonate كات الدروا. bicarbonate شو دروا bicarbonate بيسا. ياخد كبicarbonate در شو در وك أوه تشيبه وك أوه إيش هات بهتجوا. So this is through what via the anion exchanger, because bicarbonate شيكا بفريقا. So these are the mechanisms to control acidity and basicity of the inside cells. These are all ion transporters that are involved in the cellular pH regulation. The most important one, like other ion transport that other process that we discussed, the most important one is NAK ATPase. So NA pump should work in order for other transporter to function or to activate it. When NAK ATP is working, NA will decrease inside. There will be NA gradient towards the inside, يعني inside NA chemical كاتشيبه. This will act as a driving force for other transporter. When NA decrease, what will happen? NA can come in and hydrogen go out, hydrogen ion go out. So this is important in what? In cellular acidity, to decrease the acid, increase expel of the hydrogen ion. Okay, we have another transporter which is called anion exchange. This is work during cellular basicity. Why? Because it expel SiO3 bicarbonate أكات دروا كبicarbonate كردا دروا تشيبه cellular basicity كا وردا كما بيبرو تشيروا while أكاك تشيبه it will decrease the pH towards normal. But here anion exchanger. Anion exchanger present in most of the cell. Here in the cellular, during cellular basicity, it works in this direction. HCO3 out and Cl in. We have another transporters also. You can see the NAK ATP is present in almost all the cells of the body, while NaH is present in most of the cells to control hydrogen ion and pH. And we have another transporter that coupled NaH and anion exchanger. هردو أكيد يايا هردو لم transporterيت NaH وتيش SiO3Cl transporterيت يايا. So this is also worked by low NA will come in H will go out, and accompanying what? HCO3 come in and Cl go out. This is when become activated during cellular pH, during cellular acidity also. When pH decrease, pH كم بوقتها H صورة, the H should go out, the H ion should expelled, and also HCO3 come in. This is important in order to regulate the pH. This is present in some of the cells, not all the cells of the body. And in some of the other cells of the body, we have Na bicarbonate co-transport. Na bicarbonate co-transport شلون يشكل؟ The same when Na gradient, Na decrease here, there will be Na entry and HCO3 entry. This co-transport بهكوا كاركا هم Na وهم HCO3 Na تجور كواته هموي بهوي شو شون H كينا در how H expelled by the way of Na, how HCO3 come in by the way of Na. So Na gradient is very important, and Na should be low inside the cell for this mechanism to work. This is the same. This is a graph that shows how intracellular pH regulation coordinated. We have a set point here. I'm a set point here. Moving away from the set point cause activation in one of the mechanism. I'm intracellular pH. This is the central normal pH. When it go, the pH decreased like this. What will happen? Acidification activate in sodium hydrogen exchanger and cheater sodium bicarbonate exchanger. Sodium bicarbonate co-transport. Kata chieka sodium etajuro echakata daru HCO3 etajur in order to decrease this acidity. While when the set point it will go toward here, toward right, means the pH increase. What will happen? There will be alkalinization activate anion exchanger. أما تنها ميكانيزم بو basicity, cellular basicity to resist cellular basicity. چیه anion exchanger activate in which direction? In a direction of expelling HCO3 out. So this is anion exchanger. Anion exchanger called anion exchanger because it exchanged two anions but in opposite direction. HCO3 بودرويا بوناو, Cl بودرويا بوناو. So it work in either direction. Either move bicarbonate either into or out of the cell in exchange of chloride. So either alkalinizing or acidifying the cell. This according to the type of anion exchanger that present on the surface of the cell, because we have different types of anion exchanger, and anion exchange is very important. Absence or defect in the anion exchanger related to several development of several diseases. بوشي bicarbonate أو نجرينجا أو exchanger here. Bicarbonate, you know, the body need bicarbonate, the blood need and soil need bicarbonate to use it as a buffer. كاتا لقال إيش أبا combine كا وكو buffer بكاره هنري. لبرو بيرس معنا كبicarbonate كينو blood. In the renal tubule يكم جعل كدني شيء كدني فلتريا كا وحتى bicarbonate كا مو أشتنا tubule كانوا. When the bicarbonate filtered to the tubule, most of the bicarbonate reabsorbed. يعني ديسان absorption أكلنا وبكوا بنا سلكانو then to the blood. أشتنا blood because we need them. كاتا كبرناو tubule كا أروانا هلا بتشت urine زور بيتشيبه reabsorb أكلتها because we need bicarbonate. And what is this reaction? Why this reaction is so important? This equation you can see this work in both directions. So HCO3 bind to H, it will form CO2 and H2O, or H2O bind to CO2 to become H and HCO3. Of course, there is another reaction between it will become H2CO3, then dissociate to CO2 and H2O. But this is the simpler one. And this is accelerated by an enzyme called carbonic anhydrase enzyme, an important enzyme. أما بۆ گرنگە بایکاربۆنیت ناتوانێ بەناو cell membrane بڕوە. It's impermeate the plasma membrane. That's why it either should have a transporter or it should change to CO2 in order to pass through the membrane. CO2 طبعا غازة بآساني بيعروها. CO2 HCO3 equilibrium is the body's central pH buffering system. كاتما هيك كلا buffering system گرنگا كان CO2 HCO3 should be in the equilibrium state. Let's come to cell volume regulation. When extracellular osmolarity suddenly increase or decrease, what will happen to the cells? Of course, the body has several mechanisms for homeostasis of the osmolarity and solute fluid in the blood. And this, of course, if the blood maintained in normal state, the interstitium is also normal and that of the cells also normal because it prevents change. So there are several mechanisms to prevent changing in the extracellular osmolarity. But what will happen when these mechanisms couldn't be controlled, or these mechanisms couldn't control this osmotic change, or when there is something inside the cell happen and cause shrinkage or swelling of the cell? يعني shrinkage and swelling of the cell. When the osmolarity change, the cell will shrink in case of extracellular hyperosmolarity. كاتك osmolarity لێرە زۆر بوو واتە solute زۆرتر بوو لە solution، يعني ژماری سۆلیوتەکان زۆر بوو. چی ڕوایە؟ fluid لە ناو خانەکۆیەتە دەرەوە. So fluid water will come out and this will cause shrinkage of the cell. What will happen in the hypotonic solution or hypotonic extracellular fluid؟ ئەگەر hypotonic بوو osmolarity کەم بوو واتە solute کەم بوو، لەو حاڵەتەیە چی ڕوایە؟ ناو سێڵەکە solute زیاترە بێ چونکە وەختێکە وەکو یەکن there will be movement of water to inside the cell. So it will cause swelling of the cells. How the cell can resist this? The cell try to resist this and respond to this by several ion transporters also to prevent the further swelling and further shrinkage. أما طبعا زور گرنگه because if the cells shrink or swell, it will cause damaging effect to the cell. Volume regulatory increase will be activated in response to cellular shrinkage, and volume regulatory decrease activated in response to cellular swelling. كاتێکە سەڵەکە shrink بوو ئەبێ ڤۆلیۆمەکەی زیاد بکە بە پێچەوانەوە، یعنی هەوڵبازی کاتەوە و بە پێچەوانەشەوە کەواتە سەری ڕسپۆنسەکان. If you can see, what is the response to cellular swelling? The cell will activate the mechanism, so in response it should extrude ion. أبي ion كان expel كبي كاتا دروا. ion كان مانشين NaKCl. كاتا همو transporter كان activate بنكا NaOKCl أكنا دروا. When the NaKCl go out, the water go out, and this will cause decreased swelling. And in cellular shrinkage, what will happen in response to this? In order to overcome shrinkage, there will be influx of ions or solutes to inside the cell, and water follow and resist the further shrinkage and return the cell volume towards normal. These are all mechanisms to resist cell swelling. We have mechanisms and transporters that resist cell swelling, and transporters that resist, or mechanism that resist cell shrinkage. First, during cell swelling, what will happen? Of course, in both of them, one of the transport is very important, which is what? NAK ATPs. So in both mechanisms, it benefits from the gradient made by NAK ATPs, low sodium inside and high potassium inside. سود لمانا بينك تشبكة for the action of other transporter. Of course, you can see we have several transporters and not need to know every one of them, but only know that when the cell swell, the solid should go out in order for water to follow. So, and know that NAK ATPase is the driving force for that. And also know that the gradient of potassium will work to resist cell swelling, while for cell shrinkage the gradient of sodium is important. Okay, here by NAK ATPase potassium inside is high, increased. كاتا potassium زورة. كpotassium زور وشيكا there will be movement of potassium through the potassium channel, and also it will activate a transporter co-transport of potassium and chloride. كاتا potassium تشتدر chloride follow يكا بهويم transporter. Also when K go out, H will come in. This is the counter transport between K and H. Counter transport K و H. Potassium كاتدر hydrogen يتجر. Amino acid will also go out. Chloride will go out. So every mechanism become activated that goes expelling of the ions in order water follow, go out and decrease the cell swelling. But let us see why this anion exchanger acts in this way. وقلت من anion exchanger بهردو بارا كاركا. ليرا سيريكن when K go out, hydrogen come in. Hydrogen يتناول. Hydrogen need SO3 أما أبي SO3 لقلا يقبل هم تأكل وكو بفر أبي كاركا وهم قريني بو CO2 چيه بي when H come in, the anion exchanger will work in a direction of entering HCO3 أبي HCO3 بيتناو سيلكا كاتا Cl أروات دروا. This is also important, another way of extruding ions, and at the same time H and bind to HCO3 and the reaction will happen and CO2 will form. This is also the same. You can see all the solid go out and the fluid go out again, amino acid go out. These are all transporters that used during cell to resist cell swelling. You can see, as we said, anion exchange right in this direction. You can see K go out, H come in, HCO3 come in, so Cl go out, HCO3 H bind to H2CO3 and become CO2, and CO2 go out of the cell to the blood. This is explanation of these mechanisms. What are the mechanisms to resist cell shrinkage? As we said, NAK ATPase is the most important transporter that codes NA gradient. NA لنا سلكها كما بيتوا. So NA gradient, low NA concentration inside is a driving force for other transporter to work. كاتا أبتويجي NA يتناول. We have a co-transporter which is called NAKCl2 co-transporter. NAK هو 2Cl co-transporter أتوين لاين Cl2 بو أساني. كاتا أم co-transporter تشيكا. It co-transport sodium, potassium and chloride to inside the cell. When this works, this is secondary active transporter to work when there is NA gradient, NA low here. It become activated. Kata solute netajur. Why entering of the solute to the cell is important? For water to follow to prevent further shrinkage. This is one. This is the most important one during the cellular shrinkage or resist cellular shrinkage, of course, after NAK ATPase. Another co-transporter is NaCl. أمانتها NaCl. This is solute netajura. Another one is NaH. NaHtajur H there. Because hydrogen go out, look, this time the anion exchanger work in opposite direction of that of cellular swelling. لو H هاتجر بوي HCO3 هاتجر. When H go out, so the anion exchanger work in this direction. HCO3 go out and Cl come in. So Cl come in, Na and potassium come in, and the solute increase and the cell volume increase to prevent further shrinkage. This is the same. All the transporters shown here. This is explanation. Let us come to another subject, bicarbonate reabsorption by renal tubule. As we said, bicarbonate is very important and it should be reabsorbed from renal tubule to the blood. Most of it should be reabsorbed. How does reabsorption take place? And we discussed that this equation is very important. H bind to HCO3 become H2CO3 and dissociate to H2O and CO2 in order to transport bicarbonate through the cells. This is the lumen of the tubule. اما ناپۆشی تیوبیوڵەکەمانە ئەم شوێنەیە کە وردە تیوبیوڵەکە ئەڕوا تاکو چی دروستە بێ؟ یورین دروستە بێ. So this is the tubular cell. ئەمە سێڵێکی تیوبیوڵەکەیە. ئەمە چییە؟ blood vessel بڵاتی تیا. Okay. چۆن ئەو HCO3 بێنینە ناو blood. How HCO3 reabsorbed. HCO3 طبعا زۆر جا بەشوەی NaHCO3 هەمانە dissociate بۆ Na و HCO3. HCO3 bind to H to become H2CO3 and then CO2. Why it convert to CO2? Because here in the luminal membrane we do, as we said, HCO3 cannot pass through this membrane. La luminal membrane tubule مانية transporter مانية. We have no transporter of channel or exchanger for HCO3. So it should convert to CO2, then CO2 come to inside the cell. باشە سوودی چه CO2 بێتە سەڵاکە خۆیمە HCO3 مان ئەویست. Again, the reaction as we said work in this direction also. CO2 bind to H2O and it will form H2CO3 and it dissociate to H and HCO3. كواتا HCO3 بە anion exchanger مان لێرە هەیە لەسەر چی؟ لەسەر بەیزەڵی تیوبیوڵەکە أو سایدەی کە لای بڵەدا. So through anion exchanger HCO3 enter the blood and Cl come to the cell. باشە. پێم بڵێن چی وای کرد کە لێرەیە HCO3 بگۆڕێ بە CO2. یەکێ لەو شتانە چییە؟ یەکێ لەو شتانەوەی کە ئەبێ هایدرۆجین بێتە ناو ئێرە. Hydrogen should be secreted into the tubule. How hydrogen secreted to the tubule? By NaH exchanger. How NaH exchanger work? By Na gradient. كواتا أبي Na concentration ليرة كم بي. When Na concentration become low inside cell, by NAK ATPase again. بينيتان NAK ATPase چند گرنگه؟ So the last subject is Na reuptake by the kidney. Sodium reuptake by the kidney. We have several transporters that reabsorb sodium. Of course, you know sodium is very important. It creates most of the osmolarity of blood and body fluids by Na. Almost all of the Na that appear in the glomerular filtrate is reabsorbed from the kidney nephron. وكو bicarbonate sorbe امجريتها. All Na will be reabsorbed. Na filter كراوة اجتنا اما فلتريتكيا فلتر كراوة ليرة لتيوبولا كان ورده مجريتها تا هر هموي reabsorb به. The driving force for this reabsorption is what? Low intracellular sodium. Again, this is the low intracellular sodium concentration maintained by what? By NAK ATPase activity in the tubular cells. Let's see what are these transport mechanisms. Of course, renal antihypertensive therapy, part of them, which are called diuretic, are working through targeting these transporters to inhibit NA reuptake. بۆچی بزانیم ئەنتیهایپەرتەنسیڤەکان بەشێکیان بەم شێوە کارەکەن بلۆکی سۆدیۆم ڕێئەپتەیکەکەن نایڵن سۆدیۆم ڕێئەبسۆرب بێ. Why? As you know, high blood pressure there will be increased. High blood pressure بێت ئێمە چی بکەین when the pressure on the wall of the vessel increase. ئەمە یەکێ لەو ڕێگەکانە we can decrease the blood volume fluid volume كما so the pressure on the wall of the vessels decrease by زور نخاتي سر. This is one way of decreasing blood pressure. And as you know, sodium creates osmolarity. When there is sodium inside blood, it will draw water to itself. Fluid reaction. That's why they benefit from this by blocking sodium reuptake. So sodium go to the tubule to the lumen of the tubule in order to create a volume, a higher volume of urine. كواتا urine volume يكي زور درست به چونك فلودا كش reabsorb نابه when sodium not reabsorb, fluid not reabsorb, and the urine output increase. So we have condition of like edema, edema zanen accumulation of fluid in the tissue of the body, and also hypertension. In these two conditions we need what? We need to decrease the fluid in the body. This is by way blockage of one of the Na reabsorption mechanism with diuretic drug, and this is to increase sodium excretion, and water will follow and it produce hyperosmotic urine. طبعا او Na كتشت الناو tubule كان urine كوا شي درستك hyperosmotic urine which draw water to itself. So Na and water go out and increase urine output. Diuretic, why they are called diuretic? Because they cause diuresis. This is a class of antihypertensive drug. Of course, we have several classes of antihypertensive drug. Not they are all working at the same manner, but diuretic is very important because diuresis increase urine output. This is mostly used to treat blood pressure and of course to other conditions, heart failure, fluid accumulation of edema, and so on. There are three major classes of diuretic. We have loop diuretic, Butch-Pialan loop diuretic because it acts on the loop of Henle. Ama glomerulus, this is a functional unit of kidney which is called nephron. This is glomerulus. Here the filtration occur, and this is the tubule. كفلتره به لبلا دوا ماده كان يعني ناو كيوا ناو إيرها باي يورين دروز به ليرة للوبي هنليا كان transporter كمان هي كشي كاريتيكا loop diuretic inhibit this transporter. Butch-Pialan loop diuretic it act on loop of Henle. وكوشي لازكس يعني فروزيمايد لازكس تنوانه زور بستبه هاك هيك زختي برزبته هالن شي بوبكالن لازكسي بوبكا, and it increase urination frequency and amount of urination ها زوزو كساч ميزاكات. Okay, we have another drug called thiazide that act on the distal convoluted tubule, this part of the kidney, and we have potassium sparing diuretics like spironolactone and amiloride. جور diuretic لسر چی شکا لسر cortical collecting duct. Because this is important, why each diuretic act on different part of the tubule, because on different part of tubule we have different transporters. لانه هنه كان دوباره بس transporteri جواز من هي. For example, this is diuretic that act on the loop of Henle, as we said, thick ascending loop of Henle كبيلانجي لازكس or furosemide. أو نوعنا طبعا جاري كي إخوانه لكارديوفاسكولار موديول ورده لاتان آسانه به باش. اما چی من هي normally in this part of the tubule we have these transporters كشان يمانه هموي بزاني بس NAK ATPs أبي بزاني لقل چی ترمان هي NAK ATPs زورت لاي بلاده أم بشي سيلاكيه. Okay, while in the lumen چی من هي أو گرنگه بزانن we have NaKCl2. ها وتمان transporter یک من هي Na و K و 2Cl چی لیگه که transporter که بنا سیلاکه باشه. أما solutana كات now cell. This is normally in normal condition, and this is called reabsorption of the sodium, Cl, and potassium. What will happen if this blocked? Through this loop diuretic, this one is blocked, and reabsorption cannot be. So the solute will go to the lumen of the tubule and go to the urine, and it create hyperosmolar urine, and fluid will follow. Fluid diuresis. So loop diuretic block NaKCl2 cotransporter. By this it decrease NaK and Cl, and at the same time it decrease water reabsorption. Water reabsorption هي لما أنا كما كاتو أبيته هاي diuresis. Another part of the kidney, as we say, of the tubule, the drug acting on, which is called thiazide drug. A group of drug called thiazide act on the distal convoluted tubule. أم بشي كدنية ليرة سير كان شيمان هي. This is NaCl cotransporter. أما NaCl cotransporter normally چون كاركا it bring Na and Cl to inside. بيكمان بوشي Na و Cl يتناوى لو يتري شاوت من Na و K و Cl يتناوى. أما تشيا secondary active transport use the gradient of sodium, low sodium here by NAK ATPase. بيسنا كما مجرب لينو توفر بون. باش. What happen if we block this transporter? Na and Cl not come into the inside cell and it remain in the urine, create hyperosmolar urine, fluid will follow and cause diuresis. Another drug act on this part of tubule also, but on different transporter, which is NA channel. كارلسر NA channel كا بلوكيكا. وقت كي NA يتجر it prevent NA reabsorption. This is called amyloride. So thiazide diuretic block NaCl cotransporter, amyloride block Na channel. So they cause NaCl and water, decrease NaCl and water reabsorption and cause diuresis. Drug acting on cortical collecting duct. أو دراجانا چینکە کار لەسەر چیەکەن cortical collecting duct. ئەمە چیە؟ Potassium sparing diuretic. Why it has this name? Potassium sparing diuretic means it spares potassium. It prevents the removal of potassium from the body. It prevents hypokalemia. The other types of diuretic may cause hypokalemia. خۆتان زانن drug زۆری سوودی هەیە بەڵام لەگەڵ لەوانەیە ساید ئیفێکتی هەبێ ئەگەر بۆ ماوەیەکی زۆر بەکارهێنا. So the other two drugs diuretic they cause decrease in the potassium. هنا KCl2 potassium هێنا ژوورەوە بەڵام loop diuretic لازێکس چی کرد بلۆکی کرد کاتە پۆتاسیۆمیش ئەکاتە دەر. So it may cause hypokalemia if used for prolonged period of time. Here the hypokalemia not happen. Why? أما بزانين لسر چی إشكا spironolactone potassium sparing diuretic. إما receptor man هي بو aldosterone. Aldosterone is a hormone in the body that causes reabsorption of fluid. Aldosterone reabsorption fluid كام. Reabsorption sodium كا potassium كا تدر. Reabsorption of sodium causes reabsorption of water also. فشما carry aldosterone. How aldosterone do that? أم صوزانة بيننا وصحة صوزانة. This is the area that aldosterone work on it. طبعا شي لقى stimulate NAK ATPase كا stimulate چی تركا stimulate potassium كا كبتشة الدروة بلام sodium يعني تجورو NAK ATPase activate كا. This is the function of aldosterone. While spironolactone act on the aldosterone receptor but blocking this receptor. كاتامو receptor كاني كالدوستيرون كاري لسركر هنا نجروي صوديوم NAK ATPase بلوكيكا. When NAK ATPase blocked چیه به این ingredient لیره دروز نابه کات صودیوم نه تجربه potassium نکات در. That's why it's called potassium sparing diuretic. Prevent potassium to go out to the urine. طبعا الدوستيرون تبان چیه که لگلیا فلوید يتجربه چونکه صودیوم يتجربه. Another drug which is called amyloride that we said act on the tubule لا distal convoluted tubule إيش كاري أكرد كاري لسر شيء أكرد NA channel. It also act on the NA channel. ليرش همان شد بلوكي NA channel لكا. So spironolactone block aldosterone, so block NAK ATPase and NANK channel, while amyloride block sodium channel. كاتما شيء كان decrease NA and water reabsorption. If you notice that we in the loop diuretic we كان بينيتان سهمي زورتر من دانا يعني greatly decrease NA and water reabsorption or other solid reabsorption. Loop diuretic is the most potent one, but as we said it may cause hypokalemia. بلام أما شيء potassium sparing. بويا كاتي بيسمان بدو درق بي أتوانين أما بكار بينين لقال loop diuretic. ئەمە هەر سەمەری ئەوەیە بەس ئاگاتان لە یەکشت بێ ئەمە لێرە هەر یەکە و بەشێکی نیفرۆنەکەیە ئەوە تا سەهمیشی بۆ کراوە خۆی هەمووی ئاوانیە ئەمە تیوبیوڵەکەیە سێڵەکانی هەمووی وەکیەکن but this is imagine each one is a part of the tubule. This is summary of what we take and thank you.